In this video, we will discuss about the metabolic alterations that occur in the cancer cells because of the change in the tumor microenvironment. And we will also discussed what are the drugs that might cause this that might reverse these metabolic alterations in the cancer cells. So we will start with this epithelial cells. Uh, Epithelial cells that uh, because of the metabolic alterations transform themselves into the mesenchymal cells, or we can say that they will transform themselves into the epithelial cells undergoes EMT transitions, that is epithelial to mesenchymal transition. So what are the changes that might occur in these epithelial cells is shown in this uh, on the left hand side. That is, these are the processes that are down regulated, such as this FN synthesis, the asparagine is down regulated, succinated down regulated, fumarate down regulated, 2HG is down regulated. And how these things mesenchymal cells can be again regain its characteristics to the epithelial cells is shown in this red color. That is, if we use these drugs, then these mesenchymal cells can again regain its epithelial characteristics, such as this CB839 isocidenib and inositidine. And the mesenchymal cells has these characteristics, that is invasions and metastasis. And this thing, and what change in the metabolic pathways leads to this invasion and metastatic characteristics is not yet been completely identified, or there is no general consensus. There is no consensus in the science community, as different peoples have different uh, opinions about that. So next we will talk about this treatment sensitive cells. These are the cancer cells that can be recognized by the treatment, and however they will become treatment resistant cells through the down regulations of these pathways, such as this A metabolism, this OX4 pathway, glycolysis pathway, FAO pathway, and antioxidant defense pathway. Down regulations of this pathway will make the cells treatment resistant. And also the lactate that are released by the cancer associated fibroblast will cause acidifications, and that will also enhance or contributes towards this treatment resistance property in these cancer cells. Along with this leptins and arachidonic which are released by adipocytes. And these treatment resistant cells, the property that acquires these treatment resistant cells is that they are resistant against apoptosis, which is one of the most commonly triggered pathway by many drugs that are used to cure cancer. You cure or reduce the cancers within the patients. So in this treatment resistant cells, they will acquire resistance against the apoptosis pathway. However, how these treatment resistant cells can again become treatment sensitive cells, they are this in some cases these drugs can be has shown probable results to reverse this treatment resistant cells into the treatment sensitive cells, that is 2DG, 6AN and AZD3965. Now so after these two topics, now we will go through the final topic that is mentioned in the figure, is about how the cancer stem cells differentiate themselves into the this cancerous cells. So or this how the differentiate cells can become the cancer stem cells. So these differentiate cells can undergo the down regulation of these pathways to form themselves with the cancer stem cells, such as this glycolysis, down regulation of glycolysis, lactate, antioxidant defense, methionine is down regulated, and ketone bodies are also down regulated. And this at the same time, these lipid droplets will also help in this cancer stem cells formations, which are uh, found in this tumor microenvironment. And these cancer stem cells, when they again then differentiate uh, into this differentiate cancer stem cells again then differentiate into their final cancer cells is by this upregulations of this RAS or and downregulation of this glycolysis and oxophos pathway. And in order to prevent or avoid these cancer stem cells, uh, in order to regain or we can say that restate these cancer stem cells into their normal differentiated stem cells, these reagents are found to be effective. These drugs are found to be effective, such as AZC3965 L-asparginase. So by using these drugs, the amount of cancer stem cells can be reduced, and the chances of this cancer stem cell chances of these differentiated cells within the host will be reduced after some time, as there are less amount of cancer stem cells. So these are some of the metabolic alterations that has occurred in the tumor cells, and these changes are mostly due to the tumor microenvironment. So if you want to understand more about these metabolic alterations in the cancer cells because of the tumor microenvironment, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.